So again, when we're looking at our reactions, okay, or our structures, we can look at the big changes, or our big changes in this case. The thing in the middle is gone. Yep. That is a carboxylate, and we removed it, so it's a decarboxylation. So we did a decarboxy. What else did we do?
Oxidation.
We did an oxidation reaction in here. Okay. For both of those things to happen, the order is important. For the decarboxylation to occur, you have to be able to stabilize, because your electrons come in, you have to be able to take those electrons in that bond and you have to stabilize them somewhere in the rest of that structure. So we highlight them in blue. Those electrons need to be stabilized somewhere in the rest of this. Okay. The only way that they get stabilized is if that purple position is a carbonyl, because then we'd be looking at the alpha position and we'd have an enolate transition state, or an enolate intermediate. Okay? So we have to do the oxidation first. Once the oxidation is done, that facilitates the kickoff of our carboxyl group. Okay? Because we're doing that oxidation, what else would we expect? We're dumping those electrons onto NAD+. So we're generating an electron carrier. Okay? It is an important reaction because we're generating those electron carriers, which then means it will be controlled through some inhibition. ATP and NADH will both prevent this mechanism or this reaction from going. Okay? NADH is one of the products of this, or immediate products of this reaction, so we get a huge feedback inhibition, immediate feedback inhibition, and then ATP will also inhibit this. And that's because one of the products of this is NADH, and NADH, we'll find out later, goes through to make ATP. Okay? So we see kind of two feedback loops. One that's almost an immediate feedback, shut the reaction down, and one that's 6 or 7 steps further on down the road that feedback loops. Okay? Once we have our alpha-ketoglutarate, we'll convert that into succinyl-CoA. And when we take a look at that, what changed? Okay. We somehow got the CoA there, and you're saying the sulfur is actually part of CoA. The only reason it's being shown as an extra kind of bond from the CoA is we're trying to emphasize that it's a sulfur carbonyl linkage there. Okay. So the sulfur is part of CoA. So that needs to be somehow involved. So clearly one of our starting materials will be coenzyme A. That needs to be there. Okay, what else changed? We did a decarboxylation. Okay, so we have another decarboxylation. Okay, so a decarboxylation and the introduction of our coenzyme A. Okay, so lots of little nifty stuff going on with this one. To go through and do this, because it is a dehydrogenase, it's a little bit less obvious in this case. We are also producing NADH. Okay. Not super clear, but we will need NADH to go through and do this reaction. So that decarboxylase is our tip-off in this case. Okay. So both the alpha-ketoglutarate and our pyruvate, and for that matter, the previous one, our other dehydrogenase, are big, massive complexes. Okay? This is the one we've already talked about. You've got thiamine, you've got lipoic acid, you've got FAD, and you have NAD, all being involved in this. Okay? What we're trying to see in this next figure is the couple between FAD and NAD. Okay? And seeing that relationship on how we're transferring hydrogens back and forth between them. Okay? Remember, why do we have both FAD and NAD?
FAD is on short distance.
FAD is a very large molecule, so it can stabilize things relatively well by being large, so it can stabilize lots of intermediates. The issue with that is it can't move. Okay? So because it is relatively pinned, we need something a little bit more mobile. That's where our FAD can come in, or sorry, our NAD can come in and act as our electron carrier, okay, over larger distances. So we've got those big, huge complexes. After that, we'll take a look at our thioester bond of succinyl-CoA, and we'll convert that down to succinate. Okay, so what things changed here? What's that? We can say hydrolyzed, so we're losing the coenzyme A, and we're doing that, okay, by having some oxygen come in, attack, and then ultimately kick that piece out. Okay. If we go through and look at the product, we could then say, well, that very clearly shows that we have our oxygen there. If we clean that up a little bit so that we can make that stand out, really what we're saying, so we have something like that. So we've replaced the coenzyme A with an oxygen. Okay. So now the question is, what is that oxygen? Okay. Said another way, what are the things attached to it? Put R groups on our starting material. What if they were hydrogens? Would this reaction still work? Would we still produce our succinate carboxylic acid at the end? Yeah, we would. Okay, that is absolutely valid. Water, while a weak nucleophile, because we have that sulfur thiol bond in there, that is a high energy bond that can activate the carbonyl for the water attack. So that would absolutely work. Okay. The problem with that is then, does that involve GDP or the phosphate? No. Okay. So if we look at the rest of the reaction, we are also doing something with these guys, with GTP and GDP in our phosphate. Right? So that's a little bit weird. So now the question becomes is, well, if water can go through and do this, that works, and we generate a bunch of energy. Okay? But the whole point of our energy production in our reactions, because that's a big exothermic reaction, is to release that energy, is to actually do something with it. If it's just water, are we doing anything with it?
So
Yeah, if anything, it's just heating up the system.
But GTP has more energy, so it's putting the phosphate onto the GDP.
So somehow or another, I need to force the phosphate and the GDP together. Okay? But if we go through and look at our succinyl-CoA with our water, yeah, that releases energy, but it's going to release energy into those molecules, coenzyme A and potentially our succinate. Do either of those look like GDP or phosphate? No. Okay? That's a bit problematic. So we are releasing the energy, but we're releasing the energy to the wrong molecule. We need to somehow get that energy to help us produce GTP.
The enzyme does it.
Fair enough, our enzyme does it. So now what we want to do is kind of process, is there something similar to this reaction that we've seen before? Okay, where we broke a thiol bond and somehow facilitated the production of ATP. Say like here. Okay. What happened in this case? Okay. If you remember all the way back, this was back in our pyruvate conversion.
Oh, it wants the phosphate to attach by the valve, or it brings it in, allows it to.
We used a cysteine. Okay. Cysteine had which functional group at the end? Our thiol, our sulfur, which facilitated the attack here to give us that high energy bond. What then happened? The phosphate came in and attacked, kicking out the sulfur to give us the high energy phosphate bond. Okay, so the energy was used to put on phosphate that doesn't want to be on there and wants to be stable on its own with nice resonance stabilization. So we're breaking that thiol bond to set us up to put the phosphate on. Why did we put the phosphate on in the case of our pyruvate conversion? What was the very next step?
You have to kick it off
taking it off to make ATP. So when we're looking back to glycolysis, what we did in 2 steps, okay, with our pyruvate conversion, converting, what was that? Glyceraldehyde 3-phosphate all the way down to 3-phosphoglycerate. What we did in 2 steps there was generate a high energy bond so that we could dump a phosphate group on it that we could then kick off in the next enzyme. What's happening when we look at the Krebs cycle?
Because it's just not showing all those different steps.
It's not, well, so that's where we have to be careful. It's not that it's not showing all those different steps.
Other things would happen in the previous ones.
In this case, we have enzyme 1, enzyme 2. What's happening in the Krebs cycle? One enzyme is doing it all. Okay. We have, at our very first step, or actually not our very, not one enzyme, because the previous step puts on our, come back here, structure, puts on our coenzyme A.
So that's the first.
Okay, so that was the equivalent of this one, or the equivalent of the first part, doing the oxidation at that point, putting the thiol on. Okay. That was, what was that, the alpha ketoglutarate dehydrogenase? Yeah. Okay. And then in the next step, what happens? The phosphate comes in, okay, attaches, kicks out our high energy group here. Now we have a high energy phosphate bond. Okay. Now water can come in and attack, and when water comes in, it kicks off the phosphate because the phosphate wants to go back to being its free energy form. So it kicks out the phosphate with enough energy that it slams into GDP, and our phosphate goes from, yay, I'm free, to now, oh crap, I'm stuck again on something else. Okay, and that's how we end up getting our GTP. Okay, so we're taking advantage of some of the same chemistries, it's just how and when we see them. When we looked at our glycolysis pathway, it's broken down in a slightly different sequence. Okay, we never actually see the thiol bond. Okay, when we look at our Krebs cycle, it's going to advance, sorry. We actually see the thiol bond. Okay, and when we go back to glycolysis, that thiol bond is what helps put the phosphate on. And then once the phosphate is on, the next enzyme kicks the phosphate off into our ATP. It just so happens that in the Krebs cycle, we put the phosphate on and immediately kick it off. Okay. So any guesses about size, succinyl-CoA synthase, in, say, comparison to the other 2 enzymes? We've got one enzyme doing the role of 2 enzymes worth of material. It's probably going to be a lot bigger. Okay, kind of makes sense.
You're saying that
The succinyl-CoA synthase is going to be a relatively large enzyme in, say, comparison to just glyceraldehyde 3-phosphate dehydrogenase. Okay, because succinyl-CoA synthase, our synthase, is doing the role of our dehydrogenase and doing the role of this kinase. Okay, it's all being jam-packed into one big, super powerful enzyme. Okay, which we kind of talked about as being problematic, right? We got one enzyme doing everything. If it fails, we're kind of screwed over. Okay. So if we kind of ignore our pyruvate stuff, we're now at succinate. We've released GTP. GTP, like ATP, is another form of energy that can be used by transferring phosphate groups. Same general idea. Okay. So we're now at succinate. The next sequence of steps, succinate to fumarate, what happened? Structurally, what changed? This is going right to left. Yeah, I know typically you would see left to right, but I was lazy, and what this is actually a cut out of is that drawn out circle. Yeah. If we compare our structures, we have lost 2 hydrogens, which is an elimination reaction and oxidation. Okay. So if we're doing an oxidation,
Something's gotta be getting reduced.
with something got to be getting reduced, what are we likely seeing is also being contributed in this reaction?
Hydrogens to NAD.
We're probably seeing NAD+. Okay? We need something to act as those electron carriers. Okay? In our fumarate to malate, what happened? Gained. So we added hydrogen, which would suggest reduction. What else did we do?
Oxidize.
We added that oxygen, which would suggest oxidation. So is this a reduction and oxidation reaction? No. One of those reduces, one of those oxidizes, net no oxidation reduction. I think you might have said this, Jimmy, on what type of reaction it was. Maybe you didn't.
It looks like isomerization.
Fumarate to malate. And isomerization means you take the same atoms and you just move them around in different locations. Okay. Do we have just the same atoms? No. So what type of reaction happened here?
Isomerization reaction, right?
Okay, so this is where we have to be careful. So you said dehydration. Dehydration means you remove water. Did we remove all water when we went from fumarate to malate? No, what did we do? We added water, which would mean a hydration reaction. So the specifics of it would be calling it hydration because we're adding water. If we go a little bit more general and step back, what you just suggested, Jimmy, it's an addition reaction. We're adding water to the double bond. That's how we move from fumarate to malate. Kind of makes sense? And what's the very last thing that we need to form? Just curious how far you guys got through. Oxaloacetate. How do we get to oxaloacetate?
Very careful.
It's an oxidation reaction. We need to oxidize the alcohol. Okay. So we've got our malate, and we'll take that down to oxaloacetate by doing the oxidation reaction. Again, oxidation reaction, NAD. Okay. So it's looking for those pieces. Okay. Interestingly enough, this one is not inhibited by NADH, FADH, or ATP. Okay. That seems a bit odd. All of our other reactions that produced NADH were like, we want to control that. We don't want that to continue to produce NADH. So why would this one be like, actually, it's not going to be inhibited by NADH?
It's far enough along it's far enough down the chain to get there.
So that's a possibility. It's far enough through our cycle that we're like, what the heck, might as well just continue going on.
Maybe
What's
there's
in it?
something else we could do with malate once we create it, so we don't have to.
Careful. Something else we could do with what? This reaction is not inhibited. So it will produce NADH.
With the oxaloacetate.
Oxaloacetate is an important molecule. It's the beginning of our Krebs cycle, and it turns out oxaloacetate can be used in a bunch of other locations that can potentially produce something useful. Okay? Say, like an amino acid. We have our carboxylate here. We have a CH2COO minus here. What do we need to do? Turn this into an amine, and we have crap. What do we have? I have a guess.
I think it's glutamine. Or no.
I was going to say it was aspartate. We have the aspartate or glutamate. I always get them backwards. It's the number of carbons in our R group. So oxaloacetate can be used to make amino acids. Well, do we need amino acids? That's how all these things function. So oxaloacetate can be very useful because we can use that relatively quickly to go through and make an amino acid. Okay? And I say relatively quickly because we are doing a pretty big conversion through the middle there. Okay? We have to convert that down to a single bond and then do a nitrogen. Okay? So there is a sizable amount of stuff that needs to happen to that. Okay? If you think back to OChem, you might be able to come up with a relatively short synthesis to get you to that being an amine. Okay? I don't expect you to do that, but you want to process through on the organic side. Okay? So some of our reactions we do want to stop because we're producing more energy. Okay? And that's because that more energy we don't want. Okay? But there's other cases where we're producing another molecule besides our energy source that is really, really, really useful for something else. Okay? So we may want to go through and actually produce that. So what we're actually talking about now is our process of anabolism. Okay? How can we take some of these intermediates and shuffle them out somewhere else to make something useful? So when we think about Krebs cycle initially, we're just like, oh, we just need to produce our electron carriers so that we can make energy. But those intermediates we want to be able to use somewhere else. We want to actually have some kind of recycling process. So if we've produced them, I can use them somewhere else. And if I can't use them, I don't want to make them because we've got two pathways. I can produce energy. If I don't need energy, I need to do something with that molecule. If I can't do anything with that molecule, then I need to shut down the pathway if I have too much energy. Kind of makes sense? Okay. So our control points are marked nicely by our big X's. So we've got our control going immediately into the citric acid cycle. And remember the reason that we said there was that acetyl-CoA is really good at transferring carbons, the pirate form of transferring carbons. Somebody responded. Which can help us build lipids and other molecules. So the acetyl-CoA is a useful molecule. Unless I want energy, I'm going to shuttle that off somewhere else to make something else, okay, or to store my energy. So I'm going to have a big control point there. The next big control points we encounter are our alpha-ketoglutarate dehydrogenase and our isocitrate dehydrogenase. Did I get those names right? It's those two reactions. Both of them are dehydrogenases. Let's just focus on that. Okay. We want to control those points because the products coming out of that aren't as big of a deal. Okay. We can't use those quite as efficiently or quickly as we can use, say, the oxaloacetate. Okay. Which means those reactions really just produce energy. Well, if they just produce energy and I have tons of energy, I don't want to run that reaction because there's nothing else I can get out of it. You'll notice that the oxaloacetate formation, okay, the enzyme up there, isn't a control point, okay, even though it does produce energy. And we might say, well, why not? Why is that not a control point? And that's because the oxaloacetate on top of the energy is useful. So I may decide it's okay to produce a little bit more energy if I can do something with the other species. Okay? So that ends up branching, and we end up seeing a slightly different issue when it comes up to controlling. Kind of make sense?
So if you're saying, like, you have lots of ATP, then all 4 of those steps would get inhibited by the ATP?
3 steps. Okay, where the we didn't talk. I didn't mention that first one up top. So if that's the one you're counting in your 4, all the X's, yes, all the X's will be controlled or inhibited by ATP. Okay, because all of those are energy production.
So ATP is going to stop that step from happening?
Will stop that step from happening. Okay, and that's because we have tons of energy. That's why ATP is floating around. So we don't want to continue that step because what is the product of that step? More energy. Okay.
So when you get stuck then with like some alpha-ketoglutarate? Like
So we could potentially get stuck with alpha-ketoglutarate. Exactly right. Okay. So in theory, if our reaction shut down up here, or sorry, here, we would then want something to go with the alpha-ketoglutarate somewhere. Okay? And that would then go back to our anabolic methods and deciding what exactly is going on with that. And we'll look at, I think, just an image, because the textbook doesn't go into it in this chapter. And I don't think we'll talk about it in detail. But if you remember that image way back when, at the very beginning of the semester, or not beginning of the semester, beginning of this unit, I had that big massive slide with all of those spider things going on with it. Each of those spider out to something else. Okay? That's our anabolic or our catabolic processes. And so your textbook kind of addresses that in a much simpler fashion. And since you're asking, let's just kind of jump to it. Okay? And these kind of processes. Okay? So here we're looking at catabolic processes. How can we go through and make those intermediates through other mechanisms? Okay? So our focus on this slide is not supposed to be so much our citric acid cycle, okay, but is instead supposed to be focusing on these big arrows coming into the citric acid cycle. Okay? Why do we want to have alternate ways to make the molecules in our citric acid cycle? Okay, we can pick it up at a random place. What if we run out of sugar? There's no more glucose. If there's no more glucose, can we make acetyl-CoA? No. So I need to get acetyl-CoA from somewhere. Why would I need to get acetyl-CoA? Well, if there's no more glucose, I can't produce energy, and then I can't live. Okay? So we need alternate ways to make each of those molecules. Hence our catabolism. We're breaking it down. We're saying, oh crap, there's no more energy. I need to now start cannibalizing myself so that I can live, hopefully long enough that I can find a better energy source, and then I'll rebuild. Okay? So what we're looking at here is how do we get into each of those pieces. So if we look at, actually we can start with just our acetyl-CoA. How can we get acetyl-CoA? What are we tearing apart? Isoleucine, leucine, tyrosine, lysine, phenylalanine, tryptophan. What are all of those? Amino acids, which are our proteins. Okay?
That's cool. Because then
It's very cool.
It's all of these checks and balances, so when you get screwed up in one way, at least you're not going to die. There's other ways.
Exactly right. So there's an extra check within that. If we go through and look at our alpha-ketoglutarate and even our glutamate, okay, glutamate can go through to make our alpha-ketoglutarate, which is, glutamate was a derivative of our amino acids, which can be made from proline, arginine, glutamine, and histidine, also amino acids. So we can pretty much sacrifice other proteins and take the energy found in those bonds to make the intermediates within our Krebs cycle so that we can generate energy. Okay? If we can't get those molecules or those structures from our glycolysis pathway. Okay?
That's also where new oxaloacetate would come from if all that was used up somewhere else.
So if we go through and look at our oxaloacetate, interestingly enough, the only way we can get oxaloacetate is through the other molecules carrying through the cycle. Do you notice that there's any arrows pointing in at oxaloacetate? There kind of is one. We can go from aspartate across, and we can eventually make it into the oxaloacetate, but that has to go through a couple sequences. Okay. Another thing that's interesting here, why are they showing these arrows? What are they pointing at?
The place where it would enter.
What place? What do you mean?
Would show where the alpha-ketoglutarase would enter into the cycle.
Into the Krebs cycle? Okay, I can agree with that. So we are showing how it goes into the cycle, but then what's that giant pink thing? Why the heck is that there? It's the mitochondria. Where is all this happening? In the mitochondria. All of these reactions that we've talked about aren't happening free-floating in the cell. They're happening, at least in eukaryotes. I think I said that right. In our mitochondria. Okay. So if you think back again to the very beginning of our Krebs cycle, there was a little image where I showed a brief image of our electron transport chain. The electron transport chain is happening where? In the mitochondria. Okay. How did we get that to work? Krebs cycle. We needed NADH. So our NADH we said was more mobile, but where is it moving? Virtually nowhere. It's still being contained relatively tightly within the mitochondria. Okay. It can be used elsewhere, but that's where it's primarily focused. Okay. So kind of neat in that respect. Okay. So in our controls, this just summarizes what's on that previous slide with text. That's about it. Okay. So we have a control point getting into the Krebs cycle. Okay. Our on-ramp, that's our pyruvate dehydrogenase. Then your citrate synthase, your isocitrate dehydrogenase, and your alpha-keto dehydrogenase complex. So pretty much any place you have dehydrogenase, okay, not quite any place, but most places you have a dehydrogenase, you have a control point. Okay. The exceptions being out over on this other end. Okay. Those ones are not your control points. So anything before succinyl-CoA, control point. Anything after, not a control point. Okay. So we said what we could do, potential exam questions. Okay, so we have to go through and process this kind of carefully. Okay. If we are resting, we have plenty of energy. So saying a high ATP ADP ratio, okay,
Would you have a lot of?
you would have a lot of ATP because it's not getting used. Okay, it's just kind of chilling out there. Okay, so at best you're polluting somewhere evenly. Okay, if you had a higher ADP than ATP, you're not at rest. You don't have any energy. Okay, so at a resting state, we would probably expect a higher ATP, but it should be pretty even. Okay, active alpha-ketoglutarate dehydrogenase.
Probably not making it, so we wouldn't.
Okay, if we're resting, do we need more energy?
No,
No. Okay, which means should the alpha-ketoglutarate be producing NADH?
no.
No. It should be inactive. Okay, so that doesn't make sense. High ratios of oxaloacetate to citrate. And these are stupid freaking names. I'd have to go back and look at the reaction. So high ratios of oxaloacetate to citrate. So we're saying we have more oxaloacetate than we have citrate.
No, you have more citric.
Okay.
Well, isn't oxaloacetate the first? And then you should be waiting for the next?
Okay. We're waiting to trigger that next reaction. We don't want to continue through, right?
So, but you wouldn't have any oxaloacetate.
So we haven't talked about where those different molecules can end up going, so let's not focus on that end of it. Okay. Would we be processing or converting oxaloacetate to citrate?
No.
No. What happens once we form citrate? So as soon as citrate's there, it starts to disappear. But as that continues around, what happens? You form
Get back to the
oxaloacetate. So what happens to your concentration of oxaloacetate? It goes up, and your citrate
Goes down.
goes down.
But that's
Okay.
only if the cycle's going.
But it stopped, yeah. So it stopped right here. Okay. So we can't produce any more citrate. So as soon as we form the citrate, it disappears. Okay. So as soon as the citrate forms, it's now moving through our cycle. Our oxaloacetate goes up. Can the oxaloacetate be converted into citrate? No, it's shut down.
Oh, yeah, in that case.
Okay. So if that's shut down, oxaloacetate forms, citrate doesn't.
But
So,
oxaloacetate, you're saying it's just going to stay in that form? I mean, I would.
so that's where we're kind of running under the assumption we're just looking at the Krebs cycle. So just in the Krebs cycle, we cannot get rid of the oxaloacetate, so the oxaloacetate concentration should go up. Okay? So if we look at that, we would expect a high ratio. We should have more oxaloacetate than we should have citrate. Okay. Yeah. Okay. Which then said, all of them are true, right?
No,
we
B's
said
not
B.
true.
Did we say B's not true?
The dehydrated.
Oh, yeah. Active alpha ketoglutarate. Yeah, because we said that would be. We said this enzyme should be shut down, right? In a resting state, because there's no reason to produce more enzyme or more energy, so that reaction should be shut down. So that,
And you said it shouldn't be a high ATP ADP ratio,
Yeah.
it should be more equal.
so then we said just C then, right? Yeah.
But I would argue that ATP would be more than ATP, or else it won't shut it down.
But there's other things that shut it down. So that's going to be one of the things here, because, yeah, I would think that you should have a higher ATP than ADP ratio at a resting state. Okay? Because that's what's going to shut down your alpha ketoglutarate, and that's what's going to shut down the other one, which would give you their high ratio of oxaloacetate to citrate. So I have to take a look at where that question came from, and I may have had a typo on that. Or maybe I was expecting A and C as an answer. Kind of work?
Could be A and C.
Yeah. Reasonable mechanisms, sorry?
Just say, well, I guess it's hard because what constitutes a high ratio, like
Yeah.
Yeah.
2:1,
And
3:1,
you're looking at, so the other thing is that you're looking at an equilibrium system. All of it is at equilibrium. All of those concentrations are staying relatively constant. When we put it under stress, we're no longer at equilibrium. Okay? So as soon as we back off of that, we're now at an equilibrium system. All those concentrations should maintain a relatively basal level. Question 2, a little bit more difficult because we've talked about our organic mechanisms, and you might have a problem with this one, but you should be able to come up with this one. If we go back to our structures, malate to oxaloacetate, oops, we need our structure. How do we convert? So it is our dehydrogenase. What has to happen to NAD plus?
It's the hydrogen.
It has to take the hydrogen. Is it taking hydrogen positive or is it taking hydrogen negative? Hydrogen negative. Where's our hydrogen negative?
Maybe that comes from your, oh, no.
Unless that one hydrogen on the carbon.
If it came from our alcohol, we're saying the hydrogen then took electrons from oxygen.
Yeah, and it's not gonna do that. Okay.
Yeah. Okay. It's actually going to take them from the carbon. Okay. That's not an easy thing to happen, so what's going to force that to happen? Why would those electrons leave with the hydrogen? Something must be forcing those electrons off. The enzyme can pull the H plus off of our malate. That gives us the negative oxygen, which can then go in to make the pi bond of our carbonyl. Right? And then that can kick out our H minus to the NAD.
So what would you want us to,
That's it. That's what I would expect for a mechanism there.
great. So for those lacking chemistry, you just screw over.
Well, so, and I would argue no. So you need to address, and you said, okay, let's pick the hydrogen that was on the oxygen. So what you should do is test it.
It won't work.
What happens if the hydrogen takes the electrons? Okay, process through what that statement means. Hydrogen ripped electrons away from oxygen. Why should that not happen?
Because oxygen's more electronegative.
Oxygen's more electronegative. So that arrow doesn't make sense. Okay. The other part of it, look at NAD. It is NAD plus going to NADH. So you focused on the H, good part,
Let's focus on, okay.
but you also have to focus on the charge. If it was H plus coming across, that wouldn't work, so it has to be H minus. So you picked one hydrogen and said, whoops, that didn't work so nicely. Let's pick a different hydrogen. So we could pick that hydrogen, but then the hydrogen's taking electrons from carbon. That is much better than it taking it from oxygen, but still not the greatest thing. Something needs to help those electrons leave with the hydrogen. How can we do that? Well, what else changed in our structure? We have an extra bond between the oxygen and the carbon. That bond needs to form somehow. Where are their electrons? This gets us our H that becomes H plus. And the H on the other carbon is what gets us our NADH. Okay.
So we gotta
We
stick
draw
in
arrows.
some basic thing in the enzyme that's gonna pull that first hydrogen off.
So, do I expect you to draw what enzyme pulls off the hydrogen? No. Okay. If you think back to the last exam, there was a question that says, here's a mechanism, which enzyme would most likely do this? Or which amino acid would most likely represent that? Okay. So here, again, we're having an enzyme act as a base. Something relatively basic could pick off that hydrogen. Okay. So that could be our straight up basic functional groups. It could also be our negatively charged oxygens, okay, from our acetates. Or carboxylates, I should say. Does that make sense? Okay. Why use NAD plus instead of FAD? It's more mobile. Okay. Oxidizer versus reducer. Officially, that kind of question can come in here. Those are the same kind of things as oxidizing agent versus reducing agent. Okay, you got to watch out for those. If we go NAD plus, if we're going to use NAD plus, it goes to NADH. What happened to it? Okay, it was reduced, which means NAD plus is an oxidizer or an oxidizing agent. Okay, FAD, going from FAD to FADH2,
It's both.
is reduced. FAD to FADH2, what happened? You increase the number of bonds to hydrogen. It's still being reduced. Right? The both is going to depend on the direction in which you go. If we add NADH into a reaction, what is NADH going to do? Well, it'll convert to NAD plus, which means it was oxidized, which means it was acting as a reducing agent. So it's all about the direction of the reaction we're going. So if I change the question to NADH instead of FADH2, okay, well, it's still more mobile. It's still a mobility issue, but we could switch this to being asked, is it acting as an oxidizer versus something else? To really get A or B to act as a valid answer, I could say NAD plus instead of FADH2.
Gotcha.
Okay? Because then they're going at opposite directions. Does that make sense? Why is conversion of malate to oxaloacetate not allosterically controlled?
You said oxaloacetate goes into other things.
Oxaloacetate can be converted to other things relatively quickly and easily, and that's after we get past catabolism into our anabolism. Okay, and we look at anabolism, we can get lots of, come on, where's our nicer figure? Okay, well, we're not getting the nicer figure that I wanted. Oh, it's because the oxaloacetate, and the textbook doesn't do a really nice job of explaining this. Our oxaloacetate turns into ASP, which was aspartate, which can actually get shuttled out. So the issue with oxaloacetate is it can't immediately leave the mitochondria. So a lot of those particles or a lot of those molecules in our Krebs cycle are kind of trapped within the mitochondria. To get them out of the mitochondria, there's special mechanisms to push them out. So sometimes they have to be converted into something else. In the case of oxaloacetate, it has to be converted into aspartate. Once it's aspartate, then it can transfer through the mitochondrial membrane and out into free space where we can actually do something with it.
Is that
Oops.
one showing where all those pieces if it stops would go to?
Yep. So if for some reason we had tons of energy and now we had all those molecules chilling in the middle, okay, what could we do with those molecules? So in the case of isocitrate, what happens? It relatively easily passes out of the membrane. We started with isocitrate in the middle and we look outside, and it immediately goes out to isocitrate. Okay? So it can shuttle in and out of the membrane relatively easily. Once we have isocitrate, interestingly enough, what can we convert it to outside?
You know,
Is it the same thing?
Alpha-ketoglutarate.
Converted on the way in?
Yeah, so it's pretty much the reverse reaction. We end up getting the amino acids that can come out of it. Okay. Let's see, did I actually want anything interesting in any of this? No, there's just a bunch of facts. Which is pretty much just saying, with extra pieces, or no glycolysis, we need to generate the molecules to generate our energy. To do that, we have to start destroying things other than carbohydrates. That's catabolism. Okay? So that's where we're getting our proteins and our fatty acids. Okay? In anabolism, we're building up. So now we have tons of energy, too much glucose, too much energy. We need to do something with these molecules other than producing just raw energy. That's where we'll start building things up that we can then build turrets and fun things like that. You want to take it back to all those fun games that you can play on Facebook.
Are you gonna know this glyoxylate cycle?
No. Not for this class, at least. We covered the basics of what I want out of the Krebs cycle, and that's about all I really want. Okay. So lots of different reactions can happen. We will potentially talk about gluconeogenesis because I've noticed that other campuses seem to like talking about that, so I'll try and build up a quick lecture on gluconeogenesis after this exam, but I'm not super concerned about it. Okay. Yeah. So the lipid anabolism, we already talked about this actually with our acetyl-CoA. The acetyl-CoA can be used to make lipids. So lipid anabolism, making of synthesis of lipids. Okay, we do need to talk about that after the exam. Again, you'll have another, hopefully, quick lecture looking at that, but it's starting with our acetyl-CoA. And then everything that can possibly happen with our anabolism on building things up. Okay, where do all those arrows end? At amino acids. So when we're looking at our anabolism, pretty much everything is going to the synthesis of amino acids. There are a few cases where we're seeing fatty acids, which we'll have a lecture on our fatty acids, which is really lipids. Okay, and I might talk about steroids, but I kind of doubt it. Okay. The link to oxygen. So the whole point of this is looking at respiration. Okay, right? So we are producing CO2 in a lot of these individual steps. So we're exhaling these carbon molecules. Okay, well, if this is all respiration, when we think about respiration, we think about oxygen. How many times has oxygen showed up in this reaction?
Not any yet.
Not at all. It hasn't happened. And yet when we think cellular respiration, we think oxygen. It's really, really important. Why the heck is it not shown up yet? Okay, it hasn't shown up yet because oxygen is a high energy molecule. Okay, once we activate that, we need to activate it in such a way that it doesn't explode. Okay, if you take oxygen and just ignite it, it becomes a problem. Okay, that's why wood burns. Oxygen is facilitating that combustion reaction. If we just combust oxygen inside us, that's going to suck. Okay, so we need to be very, very careful about how we release the energy from our oxygen bonds. Okay, to do that, that's what our electron carriers are for. That's our NADH and our FADH2. Okay, those electron carriers transport themselves through the electron transport chain. We use the electron transport chain to slowly and carefully react oxygen. Okay, and that's where our respiration comes from and our link to oxygen. Okay, sorry, I just freaked out. Let's come back here. Okay, so that's our chapter 20. Okay, I think I want to try and keep this as just kind of a superficial overview, so if it looks like we're getting too nitty-gritty, we'll avoid that. So, big summaries, we can kind of highlight the big ones here. We're looking at 4 very large membrane-bound complexes. Okay, membrane-bound, where's that membrane? We're looking at our mitochondria. Mitochondria, close enough. Okay. And you'll notice that they're also mentioning some electron carriers. Well, didn't we have electron carriers already? Yeah, we had NADH and FADH2. And yet, what do we end up doing? We end up introducing 2 more electron carriers. Okay. Coenzyme Q and cytochrome C. So our NADH will come, and it's just NADH, NADH and technically FADH2, will come in and transfer electrons through those membrane-bound complexes to coenzyme Q and cytochrome C. Those molecules then help transfer those electrons through to eventually react with oxygen to produce water. Okay. The whole reason of doing this is really to generate a massive battery, okay, which seems a little bit weird, but that's all we're really doing. Okay. We're taking our membrane, and with all of this electron shuttling, is that we can then use that to transfer hydrogen ions from one side of the membrane to the other side of the membrane. Okay. Which then means on one side of the membrane we have a surplus of H+, and the charge becomes very, very positive. On the other side of the membrane we are missing a bunch of these H+, which means the other side becomes negative. What we've created here is technically a battery. Okay. Now what the cell does is build a system to allow that charge to equilibrate, very controlled. That charge equilibration results in production of energy, which we use as ATP. Okay? So we are building a battery to then forcibly and physically make ATP, okay, which is kind of a bizarre concept. We're actually putting the molecules together to make a bond. It's not now just launching particles out with enough energy. It's, here's these 2 particles, work nice with each other. You're not working nice, I might keep pushing you together until you form a bond. Okay, you formed a bond, I'll let go of you. Okay. So it's kind of an interesting process. A lot of the steps get very convoluted in where the electrons move because we haven't fully resolved all of those individual steps, and it is very complicated on how we transfer those electrons through. Okay. For instance, okay, if we think about our general infrastructure for transferring electrons as humans, where do we transfer our electrons? That sounds kind of weird. Electricity. Through what? That's how biology does it. How do humans transfer electrons? Through wires. Okay? Going out to the massive scale that we worked with, we transfer electrons through metals. We use metals that conduct electricity. Why do we not use nonmetals? They don't conduct electricity. Okay? Why is this now a problem for biology? Almost everything in the body is made of water, nonmetals. Okay? So how can we get the electrons to move? Okay? And again, we want them to move very, very controlled. So what biology has done has built metal clusters to help stabilize and transfer those electrons. And then we're going to move those metal clusters into different locations that we can then transfer electrons through, say, electron carriers like NADH, right, that we can then shuttle out to react with other molecules like oxygen. But we have to be very careful with those processes, okay, for the same reason that humans are very careful with our processes of transferring electrons. We teach our kids at a very young age, do not stick your finger in an electrical outlet. Right? Our bodies are doing the same thing. They've built up kind of these safety protocols to trap the electrons so that we don't get shocked at the same time. Okay? So, we've got all these fun details of your mitochondria. You've got your outer membrane, your inner membrane, and then they, of course, have to come up with every name in between. Okay? So the space in between the outer and the inner is known as the inner membrane space. Okay? Within the inner membrane, you now also have a space, and we'll refer to that as the matrix. Okay? And if we look at what's happening, we are shuttling hydrogen ions from the intermembrane space, from the matrix into the inner membrane space. Okay? That generates a very high concentrations of positive charge in here. Okay? With a very high negative on the other side, which allows for our battery to work. Why do we need this outer membrane? If our battery is perfectly functional right in here, why do I need that outer membrane?
To hold the positives together, otherwise they float off.
Hold the positives together, otherwise they float off. But what's outside the mitochondria? Don't we have another big membrane for the cell on the outside? Wouldn't that hold all the positives?
But it might destroy the cell itself.
We're looking at putting a very high concentration of hydrogen ions, also known as an acid. We're making that space exceptionally acidic. If it is exceptionally acidic, what can it do with any basic functional group? React with it. If I take a basic functional group, say like an aspartate with a negative charge, okay, and I now put a hydrogen on it, what does that change?
Not negative anymore, it's neutral.
So we've gone from a negative charge to a neutral charge. What does that change? pH by definition will change because the H+.
Changing its reactivity, it's not going to be reactive.
It's changing the reactivity. So now it can no longer accept hydrogens from somewhere else. So we're going to change the functionality of our enzyme. But we're changing all of that because we're changing the intermolecular forces. As soon as the forces change, everything goes haywire. Okay, remember when we build our proteins, we talked about our primary, secondary, and our tertiary structure. And all of that was very, very dependent on pH. I remember all the way back when we talked about our proteins and we looked at what happened as you change the pH to the structure. That's relevant because it changes the intermolecular forces. If we start dumping a whole bunch of positive charge to one side of that membrane, that positive charge with hydrogens can react with virtually anything else, changing our pH, changing the structures, changing the functionality of everything. We have to keep that trapped. Okay? If our mitochondria becomes ruptured, that then leaks out and we've got a bit of a problem. Okay? Which is why when we look at our catabolism and anabolism, we have to be kind of centered very carefully around the Krebs cycle, because our Krebs cycle is all happening inside. Well, if it's all happening in there, I have to get through two membranes before I can actually make it outside of the cell to do anything useful. Okay? So there are some very careful mechanisms involved within those membranes to help shuttle not only hydrogen ions back and forth, but also the intermediates within our Krebs cycle and glycolysis. Okay, kind of a neat little process. So here's our big summary. We take our NADH, okay, from our outside molecule and we will dump that into our variety of electron carriers. What we're showing in each of those cases are our electron carriers that are located within a variety of enzymes. Okay, so we have, if I've done this right, Jiminy Christmas. We've got our first enzyme, which will shuttle electrons to coenzyme Q, which is our electron carrier. Okay, FADH2 came from where?
From the glycolysis of the citric acid.
Okay, from our citric acid cycle. The FADH2 that we talked about in citric acid cycle is actually getting dumped directly into the electron transport chain. we no longer actually need it going into NADH because that enzyme that's producing the FADH2 in citric acid is already present. It is actually shuttling those electrons not to NADH, but to coenzyme Q. Coenzyme Q is actually our electron carrier. Okay. It can then transfer electrons to cytochrome C1, okay, which is another membrane-bound protein, which then can transfer the electrons to another electron carrier. Jeez. Sorry, I didn't know I was talking so loud, dude. Which can then transfer it to our last, sort of color coding, our last enzyme within that process. So we really have 3 to 4 enzymes that are going through and processing each of these things. These are all membrane bound. Why do we want them membrane bound?
Because everything's happening
What was the goal of electron transport chain?
to transfer it from one side to the other side.
Transfer what?
Hydrogen.
Hydrogen ions from one side of the membrane to the other, which means all of the membrane or all of the enzymes involved in electron transport
Need to be on the membrane.
will need to be in the membrane so they can facilitate that transfer of the hydrogen ions through it. If they're on one side versus the other, it's very difficult to get the hydrogen ions to move through because you'd have to now introduce something else in the membrane to get it through. Okay? The carriers will move pretty much on the surface of the membrane. So they are not directly bound to the membrane. They're kind of floating very closely on the surface. Okay? You can almost look at them as, I guess, walkers across the surface of it. Okay? I don't particularly like looking at this, but those of you who like to quantify things, you can look at the reduction potentials. Okay? Reduction potentials because in all of these cases we're looking at electron transferring, which means we're looking at reduction and oxidation reactions, which means we can go all the way back to our lovely chemistry in 152 and look at reduction and oxidation potentials to see how these things are linked and how we actually get the transferring of energy to happen in a spontaneous fashion. Okay? Really the big thing I want you to see is that your Gibbs free energy needs to be negative. Ta-da! That's about it. Okay? You can go through and do calculations. I don't think I even ask you to do any calculations in homework because I don't like doing calculations. Okay? One thing to note, our Gibbs free energy needs to be negative, but our cell potential, the cell potential needs to actually be positive for it to be a spontaneous reaction. Okay? I haven't come up with an official, like this is the scientific reason on why we decided to switch the signs on that. My best guess is that cell potentials were something that were discovered independent of Gibbs free energy. Okay? We then realized that they were related to each other. Well, Gibbs said it makes sense to look at it as a negative energy, and the electrochemist said, well, I don't want to look at a negative energy. That doesn't make sense. It needs to be positive. I want positive voltages. And they yelled at each other enough and said, fine, we just won't agree. I'll call mine positive, you call yours negative, and we'll just ignore each other. And they realized they're connected to each other. So when we look at our relationship between our Gibbs free energy and our cell potentials, there's our Gibbs, there's our cell potential. How do we get the connection between our positive and our negative? Remember, there's a stupid negative sign in it. Okay? And then as far as I can tell, that is because people did not agree on how they wanted to represent their signs. I like Gibbs representation better than cell potential.
Is the cell potential like the totality of everything within
Within that reaction? No. So that depends how far you're looking. So for an individual reaction, we can look at the cell potential for that reaction. So you can look at the coupling of two things.
Okay, so I'm just looking at the actual reaction. Not,
Yeah. So if we look at the cell potential for, say, this guy, okay, we've got a lot of energy there, but it needs to be coupled with something. Okay, so if we couple it with, say, this one down here, okay, we would have to reverse the reaction, because notice the electrons are, in both cases, are on the right-hand side of the equation. right? So we'd reverse the reaction, and our cell potential would end up being some massively positive number. So why don't we just run the succinate dehydrogenase and couple that directly to oxygen? That generates a massive cell potential, which then means a crap ton of energy gets dumped into your body at a given instant. Is that generally a good idea? No. Okay, we want to step down that process so that we can still get to that point, but we want to control how it's happening. Okay, we want to go a little bit slower and more carefully within that stepping process. Right. So there's a nice pretty picture. You can see everything that's happening within it. We bring our NADH up to our first complex, which dumps the electrons to an internal electron carrier, FMN. FMN, what does that stand for? Flavin mononucleotide. Flavin mononucleotide. Does that sound familiar? Kind of like flavin adenine dinucleotide. What is FMN?
Just one nucleotide.
It's the sister compound to FAD. FAD is flavin adenine or adenosine dinucleotide, FAD. What is FMN? Flavin mononucleotide. So it's doing effectively the same thing as FAD would, it's just a little bit smaller. Okay, it doesn't have that second nucleotide attached to it. So our NADH will come up, dump its electrons to FMN. FMN will then transfer its electrons to, remember how we said we have to deal with metals? What is FES? It's an iron sulfide complex. So FMN will help transfer the electrons to iron sulfide. Okay. That will then facilitate its transfer of electrons to coenzyme Q. In the process of doing that, what can it do?
Shuttle hydrogens.
It can shuttle hydrogens across the membrane. Okay. And the net result is that we get 4 hydrogens coming across our membrane. Okay. We've started to build our potential. Coenzyme Q, so I was wrong on coenzyme Q, sorry. Coenzyme Q moves through the membrane. Any guesses on what you could tell me, some features about coenzyme Q? It is exceptionally hydrophobic, okay, because it is floating through the membrane space. We didn't talk a whole lot about membranes, but you have to remember that we have a hydrophilic head and a hydrophobic tail. So if our coenzyme Q is moving through the membrane, it had better be relatively hydrophobic. Okay? Which we could see as probably a good thing, because now where's our electron carrier?
Protected.
Protected. It's insulated within the membrane. That's not a bad idea. Okay? That can shuttle to complex 3. Okay? Notice that you get a big, massive mess of different things happening in there. Okay, you'll notice that we're seeing cytochrome B, we're seeing another iron sulfur cluster. Okay, we're seeing cytochrome C1 popping up. So you're seeing lots of electron transfers happening within that. Okay, again, details of which we don't need to really get into. It's recognizing that you're seeing electron transfers happening through that. That eventually gets dumped out to cytochrome C, which is on the, what is that, inner membrane space. So this is the one that will float on kind of the surface of our membrane. That final transfer helps to shuttle 4 more H+ across our membrane. Okay. Then what happens? Cytochrome C chugs along on our surface and makes it to complex 4. Okay, well, we have 1, 3, and 4. What happened to 2? Nope. Coenzyme Q is our shuttler between one enzyme to another.
Is
Where is
2 the one that'll take the hydrogens to make 1 later on?
Nope. 2 will still shuttle electrons. 2 is actually, we kind of Oh, that was a horrible way to draw it. Let's do it as an eraser. There we go. Complex 2 shuttles electrons to complex 3 as well via coenzyme Q. Okay. It's the same kind of idea. Complex 2 is one of the dehydrogenases. I believe that's our succinate dehydrogenase, where we generate FAD back in our Krebs cycle. Okay. Why FAD? What happened in complex 1?
It's similar to
Complex 1 has FMN in it. Complex 2 is using FAD. Okay. So they're both doing the same kind of idea. Transferring electrons into complex 3. Complex 3 shuttles the electrons to cytochrome C. Cytochrome C then goes through a big sequence of electron transfers, and at the same time will shuttle more hydrogens across the membrane. Okay, but those electron transfers are now at a proper energy level that when we react it with oxygen, we don't blow up. Okay, and we can produce water relatively easily. So that whole process of saying, okay, we need oxygen, happens right there at the end. And that's it. Okay. That last step actually does something a little bit more important. You'll notice in complex 1 and complex 3, how many hydrogens do we shift? 4. Complex 4 actually also shifts 4. It only says 2. Okay. Where's the other 2 that I'm referencing?
those two that make water.
The 2 that are making water. Remember, we're trying to make one side positive and the other side less positive. Well, in complex 4, what's happening? We shuttle 2 across, making one side more positive, and the other 2 are being used
Make water.
to make water. Okay, to reduce water, which then means that side of the membrane becomes less positive. Okay, so we're still transferring ultimately the same number of hydrogens, we're just transferring them or dealing with the charge in a different way. Okay? Nifty little process.
So that 1/2 O2, I mean, we're not saying that's a radical, are you just
So that's an interesting question. We can only do one oxygen at a time within this process. That means we've got a reactive oxygen sitting in there somewhere. Okay? Are free radicals a good thing to have floating through the cell? No. So somehow or another, that enzyme has to hold on to that oxygen to stabilize it, to allow it to not become a nasty big issue. Okay? How is it going to do that?
Enzymes super oxidized or whatever.
Our enzymes are going to coordinate the oxygen and hold on to it. I believe, and I'm not sure I have pictures on it, this was something I shuttled onto students last year to look at. It's an iron complex that holds onto the oxygen similar to hemoglobin. That'll hold the oxygen, it'll react one oxygen off, and then it holds onto the other oxygen and waits for another round. So there's our time, so I should stop talking. So we've got kind of our big picture on each of those. After the exam, we'll come back and look at some kind of more of the details on it. Really, I just want you to know your big complexes. We got 1, 2, 3, and 4. 2 we didn't really discuss, but that is our succinate dehydrogenase. I think it's our succinate dehydrogenase. Make sure. It's the equivalent of our succinate dehydrogenase. Okay. Our succinate dehydrogenase actually shuttles electrons through FAD into coenzyme Q, but that then, of course, requires another enzyme. Okay. So it is really, really closely linked to the Krebs cycle. Okay. Either way, what you are then linking it to is getting coenzyme Q as your electron carrier, okay, which we see in this one. So the questions that you'll get will be based off of here's complex 1, here's complex 3, here's complex 4, perhaps where they're tied. What goes into 3? Coenzyme Q. Where does coenzyme Q come from? 2 and 1. Okay. They all end up at 4, producing oxygen, or consuming oxygen. Okay.